So one way of doing this is by, we'll just add a column here, and we'll just call it info. So it's just information. And what we can do now is we can record things like the gene. Gene is RPOB. We can record the protein change. Change equals, and we just keep this the same. Bar. We can also record things like the allele frequency and the allele count. To allele frequency equals 167. So I just make sure we use the same separator. Because we have fields which actually have two values, we need to use a different separator. So I've used the semicolon to separate different information types. All right, let's hit OK on that. We'll do the same here. Let me just copy this. So we'll just copy the change. Allele count is actually the same. And then for here, let's copy that again. This would be just a dot, this would be 3, and this would be 1. Okay, so now that we've stored this information, so all of this information is stored in one column, we can actually delete this. Make this a bit bigger. All right, perfect. So now you can see we've got the same exact information, but we can add in extra whatever we want in here. We can start denoting whether it's snip. So obviously this one is an indel, so we can say type equals indel. So yeah, we can add in any information that we want, and the dimensions of the dataset remains the same. And that's what's important about VCF format and why it's so special. It does mean, however, that you probably, if you want to use any of these variables to filter, for example, if we want to only extract, let's just say that this is actually RPOC, for example. If we only want to extract our variance in RPOB, we'll need to somehow kind of read this column in a special way, and it's not something that you can easily do. You'd have to kind of first extract, kind of split this into a vector, extract only the gene part, and then kind of do some kind of string matching on RPOB. So we need some kind of specialist software to do that. It does mean, however, if you're just interested in filtering based on kind of samples, so we can still say, give me only things like filter based on sample 2. So I'm only interested in a position if sample 2 has an alternate allele. Very easy to do. So we can still keep filtering like that. We can also filter based on position. So this is a nice way of storing this information. There's other information that we can store, which you might be interested in. Obviously, this is based on just a single FASTA alignment, but in reality, you're probably going to have next generation sequencing data. And that's going to have lots of reads overlapping each of these positions. So some of the interesting things that you might want to record are things like the depth. So for example, this particular position, maybe it had only 20 reads, or 10 reads, or 0 reads. So we can store that information. So we can call it S1 depth. So here we can add in some numbers. This is dummy numbers. So here what I'm saying is that this particular position for sample 1 had a read depth of 10. This one had 20, this one had 5. And so now we can start to do filtering based on, we can maybe say, all right, if everything is low depth, anything below 10, we can maybe make it missing. So our missing genotype will be, again, the full stop we're using for anything that's missing. You can start to see how, again, this is changing the format of the data, so we need something different. What we can do is, similar to this, we can start storing multiple variables in the same column. So here, let's just say we can add in the depth here. Okay, that should have changed that first to text. So that would be 10. This is going to be 20. This is going to be 5. Okay. Let's just add in some extra dummies here. And actually, let's change that again to text. This is going to be 40, 50. Nope, let's do the same here. Just random numbers. Okay. Let's remove this column. So now we've stored the depth. The only thing is, if you know that this is storing the depth, then it's obviously easy to see. But if I give that to someone who doesn't know what the numbers mean, and again, in the same way that we can store an infinite amount of variables in this column, so things like the depth, the quality, anything that you want, which is sample specific, we need to know what those, what the ordering of those variables are and what the actual variables are to begin with. So what we can do is we can add an extra column. Let's just make this smaller, and we'll just call it format. So it defines the format of these variables here. And what we can do is here we'll just say exactly what these variables are. So in here, the first one is the genotype, so I'll just say that as GT. Then I'll use the same kind of delimiter, and we'll say the next one is the depth. Right, so now straight away you know that the first variable in this particular field is the genotype, second is the depth, and it stays kind of constant for all samples. So again, we can do the same here. Right, so we've added an extra column now, but it does mean if you want to add in, let's say, the quality, so we'll just define a random quality as 100. In here you can start to add in extra things. It also means that, let's just say we're all 100. Let's make this low quality because we got low depth here, so let's just say it's 10. You can actually add in particular things that are specific to a position, so you don't actually have to fill in the qual in these two positions here if you don't want to, because our format doesn't specify that it has to be kind of fully filled out. It only specifies that this format defined here is just kind of consistent across the row, but not particularly across the column. This does mean that there might be particular variables in a file which you just don't know what they mean. So for example, if I didn't tell you what DP was, how would you know that? So one way is to create an additional file which defines all of these things. So we can kind of create a sheet here, a new sheet, which says DP, what is that? That's the depth. We'll just say it's an integer. We'll define genotype. We'll say again that's genotype. That's also an integer. We'll say quality genotype. Oh, sorry, this is the quality. This is also an integer. Okay, so now we can kind of straight away, if we provide both this table and this table, you'll pretty much know what everything is. We can also define these things here, which I guess is pretty self-explanatory, but let's just add in one or two. So variant type, we'll make this a string because it's not an integer. Things like AC, allele count, alternate allele count. So now by looking at this and this, you know exactly what everything is. Obviously, we're probably, because we need a specialist tool to read this and to interpret all these values, we're probably not going to store this as an Excel file. Also, it's a kind of proprietary format, and not everyone might have access to tools which can load up Excel. So what we can do is we can copy this, just put it above here. Now that's done something strange. Let me first paste it here. What else am I looking for? I'm just going to move this up here. So now we've got our definitions up at the top. We've got our actual data. It's not perfect because it's not like a nice format. You can see that probably if we try to load this into R, it's going to scream and complain about there not being equal fields for every row. So one way of loading, or kind of loading stuff into R, is that you can ignore some lines. And so I think by default it ignores file rows in a file start with a hash. So we can just put a hash in front of all of these. And that means that if we try to load this data into R, it's going to be ignored. But if we kind of open it up and just look at it, we still can read exactly what it is. So this is basically at the point where we're almost at what's kind of a native VCF file, or a variant call format file. It's basically just a massive text file. We can just drag all of that. Let's open up a terminal. Let's call this test.vcf, but obviously it's just a text file, and plonk that in there. Save this, and that's our file. And so we can interact with this file with VCF tools, which is a file which can read VCF files. Sorry, it's a tool which can read VCF files. There are a few extra columns which are present in VCF files, and I'll load up one for you now just so you can see what it might look like. Okay, it's complaining I've actually zipped this file, which is also actually quite handy. If you have a very large VCF text file, you can just zip it. It'll decrease the storage cost. So let's just actually, let's unzip it first. And in here, let's try open this. Where is it? Let's try open it with Excel. Okay, so here you can see our definitions. Now they've used 2 hashes. This is important because we have a line down here which just uses 1, and that contains our definitions of the columns. It also contains our definitions of the sample. So in this case, there's one sample called TB Profiler. But it's just important for any software to be able to recognize what's an actual kind of informative comment and what's the first header line. But you can see it's pretty much the same. We've got our, they've added in a chromosome because obviously most organisms you might work with have chromosomes or plasmids. They've got position. They've got this ID, which again, it's just missing in ours, but in the human context, they always have this RS ID, which you can do, or you don't have to use it if you don't want to. They have the reference, which we have, the alternate. They actually store the quality as a separate column. They have this column called filter, which you can change to pass or fail. They have this info and format, which we also have, and then just the same, just the samples. So it's basically the same, adding in a few extra columns, we've got the same kind of format. And yeah, that's pretty much what VCF format is.